Hey everybody, and welcome to week 3 of Socratic Med's 15-week MCAT supplement course. My name's Chris, and today I'm going to be teaching a lecture on viruses and their life cycles. So again, just for those of you who aren't too familiar with what Socratic Med is, we're a grassroots nonprofit company that provides sensible solutions to students with disparate medical school opportunities. And there are a bunch of ways that you can keep in touch with us if you want to learn more about us. I put a bunch of our contact information down here. We have our Instagram handle, our YouTube page. You can check out our website. It's pretty cool. And the main method that we use to keep in touch with students is this GroupMe. So you can use that link to join. I will also put my email at the end of the lecture. So you could also email me if you have any questions, or if you want me to add you to the GroupMe, you can do that as well. We also do personalized office hours. We do 30-minute time slots. So if that's something that you're interested in, it's all via Zoom. You can use the link all the way at the bottom. That's our link tree, and it'll link you to our Calendly page where you can set up with a couple of our tutors. And that'll also link you to a couple of other things as well. So definitely check those links out if you're interested in learning more. And then for a personal introduction, again, my name's Chris. I graduated from Stony Brook University in May of 2019 on a pre-med track, and I graduated with a BS in Applied Math and Statistics. I took the MCAT in April of 2021, and I scored a 520 in the 97th percentile. And I did get a perfect score on the psychology sociology section. So my tutoring specialties are biology, psychology, and sociology. And we do have tutors that specialize in all of the areas and all of the different sections of the MCAT. So definitely check out that Calendly if you want some specialized help, and it's all free. So today we're going to be talking a little bit about viruses, what they are, how they're a little bit different from other small infectious agents. So on this page, I'm pretty sure at this point everybody is familiar with what that is. That is SARS-CoV-2, which causes COVID-19, and that is an animal virus of a specific class that we'll talk about in a little bit later on in the lecture. We're just going to start with a basic introduction of viruses. So what really are viruses? They're defined as obligate intracellular parasites. So that means that they really need to be inside of the cells of another organism to reproduce. So they don't have their own replication machinery. They need a host in order to function and in order to replicate. So because they can't reproduce and function normally outside of a host, they're not considered to be cells, and they're not considered to be living organisms. So, whenever you hear people talking about vaccines, for example, they'll say, oh, it's just a dead virus. That's technically not correct, because viruses aren't live. So there's not really, you can't really kill a virus because they're not a living thing. I think probably the proper term to use would be attenuated, which means that it's sort of like a weakened strain, or they'll take out the replication machinery or something like that that weakens the virus to the point where it's not able to reproduce. Those are the types of viruses that we use in vaccines. Well, certain types of vaccines at that matter. But they are not considered cells, and they're not considered living organisms. So keep that in mind when you're learning about them. That's really what separates them from other smaller infectious agents. They were first visualized using electron microscopy because they're too small to see using light microscopy. So using a regular microscope, we can visualize things like parasites. We can see bacteria, but we can't really see viruses because they are so much smaller. They could be, hundreds of times smaller than a bacteria that they're infecting. So it was really only until we developed these electron microscopy techniques, I believe in the late 19th century, that we were able to visualize viruses. So viruses reproduce by invading cells and hijacking the host's replication machinery, as we mentioned. And while they come in all different shapes and sizes, generally all viruses have the same structure. So all it is just nucleic acid encased in a protein shell that we refer to as a capsid. So it's pretty much just this genetic information surrounded by protein. And that's really all viruses are. Obviously, they have different classes of viruses that act different ways and infect different things, but a virus is just a protein shell containing genetic information. And its one function is to spread. So we'll talk a little bit about the structure of viruses. So for this first about half of the lecture, we're going to focus on a specific type of virus called the bacteriophage. And that is the type of virus, excuse me, that infects bacteria only. So I'm sure this is pretty much the representation of a virus that you would most often see in pop culture. So if you look to the right on that diagram, this is a 2D representation of the bacteriophage. So you can see over here is the capsid head. So that's the protein coat that's surrounding the nucleic acid, which is inside of it. And most viruses are classified by the external shapes of their capsids. So these bacteriophages have what we call a polyhedral head. This is just a 2D representation, but if we saw it in 3D, it would be a three-dimensional polyhedral shape. And they also do have this sheath down here, which we can categorize as helical. So because they have multiple different shapes on this virus, they're somewhat complex in structures compared to other ones. So we consider them, we consider their structure complex. Other viruses, which we'll see in a minute on either the next slide or one of the next other slides, they're simpler. They're usually one shape. So like they could be helical, kind of like the sheath, and that would be the entire virus, or it could just be like the capsid head of this bacteriophage. And the complete virus would just be polyhedral. So the fact that there are multiple different types, multiple different shapes of proteins that are coming together to form this specific virus, we would consider it to have a complex shape. Right, compared to coronaviruses, for example. So if you go back, this coronavirus is much more simple in shape. It's just, a round protein capsid. It has those spike proteins, and it's also encased in an envelope, which we can talk about in a little bit as well. So when a bacteriophage lands on the target cell, remember we're talking about bacteria because that is the species that this specific virus will infect. Its tail fibers and then its base plate attached to the cell. So it lands on the cell with these tail fibers standing up exactly how we see it here, and then its base plate lowers onto the actual cell. So when the base plate is secured into the bacteria's cell wall, the sheath will contract and it injects the nucleic acid into the cytoplasm. So it'll attach this base plate to the cell wall. This sheath contracts, and when it contracts, this nucleic acid shoots out of the capsid head down the sheath, and it's injected into the bacteria. So note that the viruses that infect animal cells will often interact with surface receptors, and they're going to fuse with the plasma membrane. This is not possible in bacteria because, remember, bacteria have rigid cell walls. So there's really no way for these viruses to destroy the cell wall without destroying the actual cell. So the way they enter it is just by injecting their nucleic acid inside, and then that nucleic acid is going to hijack the replication machinery of that bacteria, and then it's going to create more of these bacteriophages from the inside. So many animal viruses, a membrane known as an envelope surrounds the capsid. So we have a couple of different shapes of viruses down here, a couple of different classes. We have helical, which is sort of, remember, like that shape of the sheath of that bacteriophage. And an example is the tobacco mosaic virus. I actually believe that that was the first virus that was visualized under electron microscopy. But it's a much simpler shape you can see than the bacteriophage. We also have a polyhedral, which again would be sort of like the head of the bacteriophage, but it's just the bacteria, it's just the virus itself. An example of that is the adenovirus. And then we have spherical over here, like coronaviruses or the influenza virus. And again, it's the same thing. It's just a very simple spherical shape. It's surrounded by a membranous envelope over here, and then we have glycoprotein spikes that are sticking outside of it. And you could see just side by side how much more complex the structure of a bacteriophage is compared to any of the other three. So like I just said, in the animal viruses, membranes known as envelopes surround the capsid. So you may be wondering, what is the reason, what is the function of a membranous envelope surrounding that hard, rigid protein capsid? And the point of that is it allows the animal viruses to come into the cell and come out of the cell much more easily. So the membrane is created as these new viruses are released from old host cells via budding. And that is actually, we're going to talk a little bit more in a little bit about the specific life cycles that bacteriophages and animal viruses actually go through. But if you're producing, it's pretty intuitive that if you're producing viruses inside of an animal cell, we don't really have to bust through that cell wall like we would if we were talking about, like a plant cell or a bacteria cell or something like that. So the way that some of these viruses can escape sometimes without actually destroying the host cell is through budding. So they sort of just go right to the plasma membrane, it circles around them, and then they're released in this little vesicle. And the vesicle it's a phospholipid bilayer, just like the plasma membrane of the actual host cell, and it's surrounding the capsid, and we refer to it as an envelope. So this envelope, this membrane, also allows this new virus to fuse into a new animal cell. It allows it to fuse into that cell membrane so that the naked capsid can gain entry. And it does this by two ways. So it can, depending on the virus and depending on the specific cell that it's targeting, we have this membrane. If it's similar enough and if the conditions are right, it can just automatically fuse into the membrane. The membranous envelope is going to dissociate into the phospholipid bilayer of the plasma membrane of the host cell, and then it's going to release the capsid into the cell. This is also accomplished by receptor-mediated endocytosis. So those glycoproteins that we can see, for example, on the spherical influenza virus, those can interact with surface receptors of regular cells. And these surface receptors are used in everyday functioning for other options. This is just the virus that is hijacking this sort of function, and it's using it for its own benefit against the host cell's health. So it attaches to this receptor, and then it undergoes a process known as receptor-mediated endocytosis. So the cell usually does this for things that it needs. So if it's taking in nutrients or food or anything like that, but the virus is just taking advantage of this natural system. It's attaching, and then it's endocytosed, and the spherical influenza virus would be taken up by the animal cell. And it's just important to note that this is not possible for bacteria cells. This is for animal cells only, because again, we have that rigid cell wall. So there's no way that we can have this membranous envelope fuse with the cell wall, because it's just rigid proteins. And it also does not, the bacterial cell walls do not express any receptors. So again, that's why the bacteriophage has to use, a little bit more complicated of a method. It has its tail fibers. We have that base plate that goes in, and then it injects its DNA into the host cell. So a little bit different: bacteriophages infecting bacteria, and these other animal viruses that are infecting animal cells. They're a little bit different. Okay, so we're going to talk a little bit about the lytic and lysogenic cycles, specifically for bacteria cells. And then in a couple of minutes, we're going to talk about a third cycle that we sort of just touched upon, but we'll go over it a little bit more formally, that we find in animal cells. So over here, you can sort of get a little preview about how an infection could lead to the lytic cycle or the lysogenic cycle, also known as lysis or lysogeny. So we'll start with the lytic cycle, which is probably the most well-known in mainstream in pop culture, especially with the rise of the COVID-19 pandemic. You start to see a lot of information about viral transmission, viral life cycle. And in terms of COVID-19, we're usually talking about the lytic cycle. So after the virus attaches to a cell and inserts its genome, it can enter one of two cycles. The first of which that we're going to discuss is known as the lytic cycle. So when the virus enters a cell, it will usually start expressing genes that encode for an enzyme known as hydrolase. So this is basically the first thing that the virus does when it enters the cell. So because this gene is expressed as soon as the virus enters its host, it is known as an early gene. So what hydrolase does is it breaks down the cell's existing genome, so the virus has access to building blocks to replicate its own genome. So a bacteriophage, for example, is a DNA virus. If it needs to replicate its genome, what better way to do it than with the building blocks of the own cell, of the host cell? Because in the lytic cycle, it's sort of like scorched earth. We're going to take this cell, we're going to take everything that we can, drain it of its resources, and then as soon as it's done, it's going to explode and it's going to release a bunch of viruses. So the viruses really don't care about destroying the genome. So it's going to break down the host cell's DNA. It's going to use those nucleotides, and it's going to start replicating its own genome. It's going to start replicating its capsids, all of those parts, and then it's going to start putting them together. So as the viral genome is replicated, each new virus's capsid is spontaneously assembled around its genetic information. So, we have that DNA, it turns into mRNA, and then these bacteriophages are going to use the host cell, that bacteria cell's own replication machinery, the ribosomes. It's going to use those to start making its own proteins. And as these capsid proteins and all these structural proteins that make up the bacteriophage are created. They spontaneously assemble around the genetic information, and it produces, a bunch of copies of that original bacteriophage. So as the number of newly assembled viruses increase in the host cell, the viral genomes start producing lysozyme. So that's another enzyme, but it's not an early gene. It's actually known as a late gene. The enzyme that will aid in the next step of the lytic cycle by destroying the bacterium's cell wall. So now that we're inside of the bacterial cell, and we're creating more and more of these bacteriophages, remember, the lytic cycle, at the end of it, the host cell dies. So at this point, these bacteriophages are trapped inside this rigid cell wall, and they need a way to get out. So it's not enough that we're just creating more and more and more, because there's a chance that this cell might die and it'll never lyse. There are a couple of different ways that cells can die. So if that cell dies and everything inside it dies before these viruses are released, then the virus failed. It did not do its job. So what happens is these viruses encode for lysozyme as well. And as lysozyme is released, only after a certain amount of bacteriophages are created, it starts breaking down the cell wall. So once the cell wall is degraded, water continues to move into the cell. So that cell wall, it protects from outside pathogens, but it also functions to keep pressure equilibrium. So the osmotic pressure is pushing outward. As water moves into the cell, it's pushing outward, and that cell wall usually keeps the pressure inward, and it keeps that cell turgid. It keeps it very rigid. But when there's no more cell wall, water is going to continue to move into the cell, and it's going to continue to push out on the cell membrane. And the cell membrane does not do a great job of containing water. So without the cell wall counteracting that osmotic pressure, the bacteria cell bursts, it lyses, it dies, and it releases about 100 bacteriophages into the environment. So those 100 bacteriophages are then in the surrounding tissue. And if they're in a colony of bacteria, they can very quickly find another bacteria, and the process begins all over again. So they'll attach right to that wall, inject their DNA, and start this entire process over. And as I mentioned before, since lysozyme is created later in the infection, it is known as a late gene. And you can use this diagram to the left. It pretty much puts everything, summarizes everything very well. So I added that. If you want to pause and take a look at it. Step 1, attachment, penetration, and then we're transcribing. Remember, bacteriophages carry DNA, so all they have to do is transcribe that DNA into mRNA, and they're good to go. It's going to replicate, it's going to synthesize a bunch of proteins, assemble the proteins, and then boom, bursts after lysozyme. So while the lytic cycle is a very effective cycle that viruses use to spread pretty rapidly, it very quickly kills the host. So sometimes that's not really the best method. It's not really the best strategy for these viruses to spread. So remember that the viruses can't replicate without a host. So if a virus kills the host too quickly, it's not going to be able to spread. So some viruses enter what we call the lysogenic cycle. And this is a process in which the virus inserts its genome into the bacterium's genome. So it's a little bit more strategic, and it's a little bit more insidious. So after the bacteriophage's genome is inserted into the host's genome, it's known as a prophage, and the cell is now known as a lysogen. So the prophage is not expressed due to a phage-encoded repressor protein. It remains silent and is reproduced as the host cell multiplies normally. So instead of coming into the cell, it enters the cell the same way. Say we're talking about a bacteriophage and it's entering a bacteria cell. Enters the same way. It injects the DNA in, and then it starts, it injects the DNA, and the DNA is in the cytoplasm. But instead of immediately starting to, break down the host genome and start creating more bacteriophages, what it does is it enters the cell's genome without destroying it, and it inserts itself into it. And it remains silent because of this repressor protein. So basically it stays in that genome silently. The cell is not aware that it's in the genome. It doesn't detect that anything's wrong. So the cell goes about its regular business, and what does this regular cell do? It replicates. It divides its own self. So as it's creating more, as it's multiplying, this bacteria cell is multiplying, it's also multiplying its own genome. And in that genome now we have that prophage. So it is inadvertently multiplying. It's inadvertently replicating this viral DNA, although it is still dormant. So eventually the cell switches from its lysogenic cycle to the lytic cycle after the prophage is removed from the host genome called in a process called excision. So the switch from the lysogenic to the lytic cycle can be caused by a number of factors, but it's usually the result of environmental stressors like hypoxia, toxins, stress. So an example, we're talking about bacteriophage, but animal viruses also undergo this lysogenic cycle. And one of the more common examples that you may see is the herpes simplex 1 virus, which affects humans. And that is the common cause for cold sores. So I think it's a vast majority of Americans, at least, do have this provirus inside their cells. It's just dormant. It's not expressed until there are stressful conditions. And for human beings, that could be actual stress. It could be starvation, dehydration, until something triggers that. And then we get an outburst because these cells that contain this provirus, this viral DNA, they switch from that lysogenic dormant cycle to the lytic cycle due to those stressors. And then that's how we get an outbreak. That's how those viruses start to spread. So that's why viruses can remain dormant for years before they're switched over to that lytic cycle. And if you look to the right, I put a diagram. That's the bacteriophage. It gives you sort of a good illustration of how the prophage is integrated into that circular bacterial genome. So the phage attaches to the cell, enters the DNA. Phage DNA enters the bacterial cell, like we said before. And then you see on that step 3, the phage DNA integrates into the bacterial cell. So you could see that prophage that's inside the bacteria, and the bacteria is not aware. It's not aware that there's a prophage inside its genome, and it's going to replicate. So you see it goes from one parent cell to 2 daughter cells, and both of those cells have identical genomes, including that prophage. And this can go on for a very long time until they switch to the lytic cycle. Okay, so we're going to do a quick review question. Read this over. Why can't a human influenza virus infect the bacteria? You can pause the video right now, take about a minute. I wouldn't take any more than that. It's a pretty simple question. So pause the video, take a look over, and then when you're done, unpause it and we'll go over it. Okay, so why can't a human influenza virus infect a bacteria cell? So the answer to this question is A, it does not express surface receptors, because remember, we're talking about bacteria cells. They have that rigid cell wall. They're not expressing those receptors, and a human influenza virus needs those receptors in order to get in, so that won't work. B, influenza requires a rigid cell wall to gain entry. That's not true. Rigid cell walls, bacteriophages are able to get through the cell walls, but influenza is not. C, bacteria secrete toxins that denature the influenza virus. That's not true either. That's not why even if it were true, that's not really why human influenza virus wouldn't be able to infect the bacteria cell. It's really because it's protected by a cell wall. And D, influenza does not enter the lytic cycle. Again, even if that is or is not true, that's not why the influenza virus cannot infect the bacteria cell. So the only one that has true information and adequately answers the question is A, it doesn't express surface receptors for the influenza virus. Okay, so we've been talking about bacteriophages for most of this. We're going to focus on animal viruses, and then we'll talk about other infectious agents really quickly at the end of the lecture. So we will continue on. Phages versus animal virus. And phage, if I haven't explained it already, is just a shortening of the word bacteriophage. They both mean the same thing. So viruses that infect animal cells are a bit different than phages due to the structural differences between bacteria and animal cells. Recall that bacteria cells have rigid cell walls. They do not permit entry of viruses. Bacteriophages inject their genome through the cell wall, but they do not pass through it. So we've been talking about that at length in this lecture. But animal cells, however, do not have a cell wall, but they're surrounded only by a phospholipid bilayer. So remember that from your high school biology classes. Animal cells also express surface receptors on their membranes, like we mentioned. Bacteria cells do not have receptors because they have a cell wall. So because of this, many animal viruses take advantage of these receptors and enter the cell via endocytosis. Alternatively, some viruses surrounded by a membranous envelope can automatically fuse with the membrane to release their capsids into the cytoplasm. And I put a picture at the bottom so you can get a sort of visualization of how a bacteria cell and an animal cell are a little bit different. The bacteria cell to the left You could see they have those little cilia sticking out and then those two flagella at the end, and that rigid yellow cell wall protects the cell membrane and the cytoplasm from, outside stresses, and specifically certain types of viruses as well. And then you look over to the right, the animal cell over there does not have a cell wall. It's only surrounded by a plasma membrane. And if you actually look to the bottom right, you can sort of see a sort of budding inward of that membrane. And that's really what it would look like if the virus were taking advantage of endocytosis or fusion. So it would sort of bud out. You could see that little pinching of the membrane. The pinching area is going to come together, and we're going to have a little vesicle that is inside of the animal cell. So that's how viruses gain entry into animal cells. So animal viruses may also enter lytic and lysogenic cycles after infecting a host cell, very similarly to how bacteriophages do it once they're inside of that bacteria cell. When a viral genome is inserted into the host genome during the lysogenic cycle of an animal cell, it is known as a provirus. So I used the word provirus a couple of minutes ago. When we're talking about a bacteriophage in bacteria, entering the lysogenic cycle and they're inserting that genetic information into the host cell's genome, that specific part is called a prophage. When we're talking about animals and animal cells entering lysogenic cycles, the genetic information is called a provirus. So it's very similar. It's the same concept. We just use different words because it's an animal virus and it's not a bacteriophage. So we say provirus. So those are pretty much exactly the same, the lytic and lysogenic cycle. Animal cells, though, can actually enter a third cycle, which is known as the productive cycle. So the productive cycle is sort of like, it's sort of like a slightly altered lytic cycle. It's almost identical, except at the very end. So instead of lysing the cell, the cell remains intact. So the viral cell inside increases, but because there's no cell wall, these viruses, remember, they came in via fusion of the membrane, or they came in via receptor-mediated endocytosis. They can actually exit the cell in the same way. So if they have that envelope, it's very easy for them. Not even before they have the envelope, it's easy for them to bud out of the membrane. And when they bud out of the membrane, that's what actually forms their envelope. So if you look to the right over here, this describes everything. So over here at 1, you could see the actual endocytosis when it's going in. You can take a look at this on your own if you'd like, but when we come up here to 6, you can actually see it budding outward. So you can see how this membrane actually starts to form around that protein capsid, and it's eventually going to push outward, pinch off, and it's going to be surrounded by that own plasma membrane that was made up of the original cell membrane. And that animal cell, it's designed to do that. So it stays intact. It doesn't lyse like a bacteria would at the end of a lytic cycle. And that's pretty advantageous because the host cell remains intact and it's actively producing new viruses they are released. So remember, with the lytic cycle, once you have 100 or so bacteriophages, that cell is going to burst and it's not going to be able to produce anything else. But with this productive cycle, and we call it productive because it is productive compared to the lytic cycle, we can just keep on using this animal cell for as long as we possibly can without lysing it. Okay, a little bit about HIV and retroviruses. HIV, which is the human immunodeficiency virus, affects the immune system of humans. And I just put this on here really quickly because when we talk about viruses on the MCAT, they very much like to talk about HIV, hepatitis, but HIV very specifically, I'll see a lot of passages regarding. So it's very good to have a pretty decent background in this, even though a lot of times we will see it in context of passages. On the MCAT, it's a good idea to have background information about it. So if you don't already know, HIV affects the human immune system. It expresses a protein that can interact with the CD4 receptor on human T cells. So remember, we talked about that receptor-mediated endocytosis. Those CD4 receptors are used in everyday immune functioning, and the HIV virus has found a way to sort of hijack this system and gain entry into the human T cells where it destroys them and multiplies. So as the viral count increases, the white blood cell of the individual is going to decrease. The individual becomes susceptible to other infections, and these infections severely debilitate and they eventually kill the individual if the individual is not being treated. I mean, nowadays we have a lot of different treatments for HIV. We have like antiretrovirals. We have a bunch of different treatments. There are constantly like new studies being performed on different types of treatment, different cures. So it's really interesting. If it's something you're interested in, it's definitely definitely worth looking up. There's some really cool research out there on HIV prevention and curing, actually. So HIV is classified as a retrovirus, and a retrovirus is a virus with an RNA genome that undergoes reverse transcription to integrate viral DNA into the host cell. So that's sort of part of the lysogenic cycle. Remember, with the bacteriophage, it's a DNA. It's a DNA virus. So it injects its DNA directly into the cell, and then that can be automatically integrated right into that cell's genome, right into the bacteria cell's genome. But if we have HIV, which has an RNA genome, and we infect a human cell, we can't integrate RNA into a DNA genome. It just doesn't work like that. So these retroviruses have come up with a way to change their RNA into DNA and then insert it into the host genome. And what they do is they encode for a protein that we call reverse transcriptase, which is also known as RNA-dependent DNA polymerase. So what that does is it takes RNA as a template and it creates DNA, which obviously is opposite from the central dogma. It always goes DNA, RNA, not always, but mostly DNA, RNA, protein. So this reverse transcriptase allows them to sort of defy that central dogma, create DNA from their own genetic information and integrate it into the host cell as a provirus. And that's one of the super tricky things about HIV. So we have a bunch of pre-exposure prophylactic medicines, and what that does, they're basically reverse transcriptase inhibitors. So if you're taking this constantly before HIV infection and you come in contact with the HIV virus, the reverse transcriptase is not going to work initially enough to create enough DNA to integrate it into the host genome. The problem is that only works before the HIV is able to create DNA and integrate it. So once that DNA is integrated into cells, it is much, much, much more difficult to get rid of those genes that are encoding for this HIV infection. So that is one of the hot topics of research too, is how is it possible to target cells that have this HIV provirus in it? How do we get rid of it? Because like I said, once it's in there, it's very difficult for the cells. It's very difficult for your immune system to even notice anything is wrong until these actual that part of the genome is actually being expressed and creating that virus. If it's not creating the virus, it's very difficult to see. So positive and negative RNA strand viruses. Viruses with single-stranded RNA genomes can either be positive or negative strand. Positive strand RNA viruses carry a genome that act directly as mRNA in the host cell. So the DNA, the RNA that they carry can immediately be transcribed. It can go into the cell, and the ribosomes of that host cell are going to read this RNA, and they are going to immediately start transcribing those proteins. For this reason, we refer to these genomes as infective. So positive sense RNA must encode RNA-dependent RNA polymerase because animal cells do not create RNA from other RNA templates. Again, remember the central dogma. So retroviruses, we have RNA-dependent DNA polymerase because it's using RNA to create DNA. And now for these sort of viruses, we're using RNA-dependent RNA polymerase. We're creating RNA from other RNA. And again, human cells don't do that. We create RNA from DNA. So we don't encode for this gene. So these viruses have to, if they're that positive strand DNA. So RNA-dependent RNA polymerase must first create a complementary strand to use as a template before replicating the infective RNA from its genome. Because remember how polymerization works, it's all complementary. So if we create a complementary strand from that positive strand RNA, and we try to make a protein from it, it's gonna code for, a useless polypeptide, basically. It's not going to do anything. So that's why we have to create that complementary strand from this positive sense, and then those complementary strands can create more of those positive strand RNAs, and then that's how these viruses create RNA. So viruses that cause the common cold are examples of RNA viruses, of positive strand RNA viruses, excuse me. So on the other hand, we have negative strand RNA viruses that carry the template strands to create mRNA. So the positive strands, that RNA genome can immediately be used as mRNA in the host cell. The negative strand RNA viruses only have the complement, that template strand. So they need to also use RNA-dependent RNA polymerase, but they actually have to carry that protein. If they only encode for that protein, there is no possible way that it can be created when it enters the cell, because all it has is that complementary negative strand RNA. So if we have no proteins and just that RNA, there's no possible way that we can make that complementary strand, because our host cells, our human cells, do not code for RNA-dependent RNA polymerase. So these negative strand RNA viruses have to have this protein already created in order to be able to function and create more. Right, because the genome can't be translated to usable polypeptides, they have to carry the RNA-dependent RNA polymerase. And examples of these RNA viruses are rabies, measles, and influenza. So if you want to take a minute as well, you can look to the right. There's a little bit more information about positive sense RNA viruses and those negative sense RNA viruses. And we'll do one more review question. So again, which is true about negative strand RNA viruses? Pause this, take a minute, write down your answer, because I'll go over it in just 1 second. Okay, so which is true about RNA viruses? Negative strand RNA viruses. So the correct answer is C. They both encode and carry RNA-dependent RNA polymerase, like we were just talking about. So A, they encode for RNA-dependent polymerase but do not carry it. As I just explained, that would not work, because if they don't carry RNA-dependent RNA polymerase, they would have no way to start the process. There's no possible way to start copying their genome, so they wouldn't be able to replicate. B, they carry RNA-dependent RNA polymerase, but they do not encode it. Doesn't really make too much sense, because in order for them to carry it, they would have to encode it. So that was more of a distractor. Because again, these host cells do not create RNA-dependent RNA polymerase, so it has to be the virus that's encoding. C, as we said, they both encode and carry RNA-dependent RNA polymerase. That's correct. D, they contain an infective genome, so that is false. Remember, positive strand RNA viruses are the ones that are considered infective genomes, because as soon as they're injected into the cell, they can be used immediately as mRNA. All right, and we'll talk a little bit about other infectious agents. That is a picture of, I believe those are the prions in an E. coli cell. Okay, so first we'll talk about viroids. Viroid is a singular piece of single-stranded RNA, a circular piece of single-stranded RNA, excuse me. So they usually do not code for proteins. They're usually enzymatic by nature, and they lack capsids. So if you look to the right, this is a picture of a viroid. This is actually one single continuous strand of RNA. So it's actually looping around and it's connecting at different points with itself. So they're usually 200 to 400 bases long, and they have large regions of self-complementarity, which is the ability to pair bases with itself, and that's exactly what it does. So any of these circular white regions, they're not paired, but anytime you see these horizontal bars, this horizontal banding down, those are areas of self-complementarity where these bases are attaching to themselves on the same single-stranded RNA. And this provides a little bit more stability for the viroid, because remember, RNAs are very sticky. They're single-stranded and they have very sticky ends. So it's not completely paired with each other, and there are some regions over here and over here, obviously, where they're not complementary. So that's basically what a viroid looks like. They're mostly implicated in plant diseases. I think just only recently there was one pathogenic viroid that was identified in humans, and it's the one that causes hepatitis D. So viroids they don't contain capsids, and they don't really code for proteins, so they're not competent in replicating themselves. So they basically have to piggyback off of other viruses. So hepatitis D, for example, they can only enter liver cells by co-infection with hepatitis B. So they'll actually enter the hepatitis V capsid and then basically just use hepatitis B to reproduce themselves. That's really all in terms of the MCAT that you really need to know about viroids. They don't pop up very often. They're super interesting if you're interested in virology, if you're interested in botany also. Definitely give them a look. But in terms of the MCAT and the scope of the MCAT, this slide is pretty much all you'll really need to know for them. They don't pop up too often. We also have prions, and those basically, they're just self-replicating defective proteins. So self-replicating means that proteins are directly producing other proteins. And again, you could probably see a sort of theme that's going on with these sort of microscopic infectious agents is that they're just really not following the central dogma that we've always been, we've always been taught, how DNA goes to RNA, goes to protein. So like in this case, proteins are self-replicating themselves. And the reason why they're able to do this is because all they really are, all these prions really are just misfolded versions of proteins that have already been produced. So proteins that you would find naturally accidentally get folded the wrong way. If it's genetic or if it's transmitted from another organism, when the prion comes into contact with a properly folded protein, it acts as a template and it alters the normal protein into a defective form. So based on any of the forces that we talk about when we're talking about protein folding, we have hydrogen bonding, ionic bonding, disulfide bonds, all of those, van der Waals interactions. It induces this properly folded protein into this misfolded form, and it's defective and it's dangerous. So prions are responsible for diseases in mammals known as transmissible spongiform encephalopathies, or TSEs for short. And these are usually degenerative diseases of the nervous system. So we can usually see this in the brain and the central nervous system, usually the brain and the spinal cord. And really more advanced stages, when you take pictures, using various imaging techniques, you'll actually start to see holes in like visible holes in the central nervous system. And that's why it's so degenerative. So in humans, the disease caused by prions are very rare. It strikes about 1 to 2 people per million people. It's a lot more common in, say, bovines or cows. Mad cow disease is a disease that's formed that is prions are responsible for it, and it's the same exact thing. It is a degenerative disease of the cow's nervous system, and it's due to a buildup of these misfolded proteins. One, because the buildup of the proteins is interfering with daily functioning, and 2, because there is a lot less of normally folded proteins, and those proteins are not doing their job. So it can cause neuron death, and this neuron death will lead to loss of coordination. It'll lead to dementia and eventually death. So it can actually take decades for humans to exhibit symptoms of a disease caused by a prion. So it's very quiet. And parasites. So parasites are a little bit larger. They're not subviral. The other two, the prions and viroids, are considered subviral agents, but parasites are a little larger, but they're also much more common than viroids and prions, and I definitely wanted to touch upon them. So parasites are really just organisms that live on or inside a host at the expense of that host. I'm sure we're all familiar with them. Per the CDC, there are 3 classes of parasites. So protozoa are the first type. They are microscopic, pretty small. They're single-celled organisms that may be parasitic or non-parasitic. And the parasitic ones generally inhabit the digestive tract or the blood and tissue of humans during an infection. And they're cured with antiparasitic or antibiotic medications. Helminths are parasitic worms. That comes from the Greek word for worm. And they are visible to the naked eye. So these are big worms that are parasites that, they infect humans, they infect dogs, household pets, if you ever had them. I'm sure a lot of people have had experience with, a pet having worms. So they also may be parasitic or non-parasitic in by nature, but they do not multiply in their human hosts. So once a worm is in there, it's just going to grow and grow, but it's not going to multiply inside a human body. So they're mostly found in the GI tract, but they can also be found in the blood, lymphatic tissue, and some worms can even be found in subcutaneous tissue by your skin. So ectoparasites are the last class, and these are the larger organisms. They're such as ticks, louses, fleas, lice. So these are the ones that attach or burrow into the skin and remain for relatively long periods of time. Some people may broadly use the word ectoparasite to describe, arthropods like mosquitoes, for example, but they're not generally considered to be ectoparasites because there is not a long period of time which they are attached to the human. So they do a mosquito, if we're talking about, for example, they will take your blood, it will feed off of a host, but we don't really consider them parasites because they're not attached for a long period of time. Whereas ticks or fleas, they could be attached for weeks or even months. And that brings us to the end of the content lecture for today. I hope this was helpful. It's not going to pop up too much on the MCAT, but it's really important that you know this stuff. Background information will never, ever be a bad thing, especially when it comes to reading those passages. If you noticed any mistakes, if you have any questions about what I talked about, if you want to be added to the GroupMe, you know, any general question about the MCAT, about prepping for it, please don't hesitate to email me. Again, my email is right there. Other than that, happy studying, and I will see you guys next week.